Welcome to the intriguing realm of cell biology. Today, we will explore the complex process of translation, which synthesizes proteins from messenger RNA molecules. This vital process shapes cellular function and outlines an organism's genetic makeup. Join us as we delve into ribosome profiling, synthetic biology, and translational control. Let's begin. Ribosome profiling is a technique used to study ribosome positions on mRNA molecules. It helps identify translation initiation sites, ribosome pausing, and stalling. This technique involves lysis of cells, RNA digestion, isolation and sequencing of ribosome-protected fragments, RPFs. Researchers can analyze RPF profiles to understand translation efficiency and gene expression regulation. In the video, we discuss codon usage bias, which is the preference of certain codons during translation. This bias is influenced by factors like mutational bias and translational selection. It affects protein expression and folding, and understanding it is important for various applications in protein research. RNA molecules undergo various modifications that can impact translation efficiency and accuracy. One commonly studied modification is RNA methylation, where a methyl group is added to the RNA molecule. RNA methylation plays a crucial role in regulating gene expression by influencing RNA stability, RNA-protein interactions, and translation initiation. Other RNA modifications, such as pseudouracilation and N6-thronylcarbamoyladenosine, T6A, modification, also affect translational processes. Researchers are actively investigating the functional roles of RNA modifications and their impact on translation and cellular functions. Non-canonical translation refers to translation mechanisms that deviate from the standard process involving ribosomes scanning mRNA molecules and translating protein coding sequences. One such mechanism is internal ribosome entry site, IRES-mediated translation, where ribosomes directly bind to specific sequences within mRNA molecules and initiate translation independent of the 5-cap structure. Non-canonical translation is prevalent in viruses and certain cellular processes, playing a crucial role in regulating gene expression during stress, viral infections, and cell differentiation. Riboswitches are regulatory RNA elements present within mRNA molecules that directly bind small molecules, such as metabolites or ions, and control gene expression. They function as genetic on/off switches by undergoing conformational changes upon ligand binding, which either promotes or inhibits translation initiation. Riboswitches are involved in diverse cellular processes, including nutrient sensing, antibiotic resistance, and stress responses. Studying riboswitches provides insights into the intricate regulatory mechanisms that control translation at the RNA level. Translational frame shifting is a phenomenon in which the ribosome shifts reading frame during translation, resulting in the synthesis of a different protein. This mechanism is utilized by certain viruses and cellular organisms to regulate gene expression and generate unique protein isoforms. Frame shifting is often mediated by specialized RNA structures or specific sequence motifs that cause the ribosome to slip or hop to an alternative reading frame, leading to the production of distinct protein products. Epigenetic modifications, such as DNA methylation and histone modifications, play essential roles in gene expression regulation. Recent studies have also highlighted the importance of epigenetic mechanisms in controlling translation. For example, changes in DNA methylation patterns can affect the accessibility of mRNA molecules to ribosomes, thereby influencing translation rates. Histone modifications can also indirectly impact translation by regulating chromatin accessibility and gene expression. Understanding the crosstalk between epigenetic modifications and translation provides critical insights into cellular development, disease progression, and therapeutic interventions. The Michelson-Morley experiment in the late 19th century aimed to confirm the existence of the luminiferous ether, a medium believed to convey light waves. Using an interferometer to study the interference patterns of split light beams, the experiment unexpectedly found no evidence for the ether. This challenged classical theories and paved the way for Albert Einstein's special relativity theory, transforming our comprehension of time, space, and light. Ribosome biogenesis is a complex and highly regulated process that involves the assembly of ribosomal subunits from multiple ribosomal RNA, RNA, molecules and many ribosomal proteins. It occurs in a specialized subcellular compartment called the nucleolus. Ribosome biogenesis is tightly coupled with cell growth and proliferation, and any defects in this process can lead to cellular dysfunction or developmental abnormalities. By studying ribosome biogenesis, researchers gain insights into the regulation of protein synthesis and its implications in various diseases, such as cancer and ribosomopathies. In the video, we will explore the intriguing concept of mRNA localization and translation. Recent findings challenge the traditional notion that mRNA is confined to the cytoplasm. Instead, Specific mRNA molecules are now known to be localized in different cellular regions, allowing for precise control of protein synthesis. This localization is achieved through the recognition of mRNA sequences or RNA-binding proteins, which facilitate mRNA transport and translation at specific sites. Understanding this process is essential for comprehending cellular development, synaptic plasticity, and related disorders. Advancements in computational biology and high-throughput sequencing technologies have facilitated the in silico analysis of translation. Researchers can now analyze large-scale datasets to explore translation dynamics, codon usage patterns, and identify regulatory elements within mRNA molecules. Computational tools and algorithms have been developed to predict translation efficiency, identify translation initiation sites, and study the impact of mutations on protein synthesis. In silico analysis of translation complements experimental approaches, providing a comprehensive understanding of translation regulation and dynamics. In the video, we will explore how synthetic biology integrates knowledge from biology, engineering, and computer science to create artificial biological systems. Specifically, we will look at how synthetic biology enables us to engineer gene expression circuits and manipulate translation processes. By designing synthetic RNA elements, riboswitches, and modified ribosome binding sites, researchers can control translation rates, regulate protein expression levels, and construct intricate gene networks. The applications of synthetic biology extend beyond basic research, offering exciting possibilities for biotechnology and medical treatments. In conclusion, protein synthesis is a complex process influenced by various factors. From studying ribosome profiling to synthetic biology, we gain insights into cellular function, diseases, and potential therapies. Let's continue exploring the secrets of cellular machinery together. Thank you for joining me in this journey through cell biology and translation.